Good afternoon, everyone. At Cullinan, we're very excited because we have a very catalyst-rich 2026 with multiple milestones throughout our portfolio, which has really been enabled by our pipeline strategy, and that's what I'm going to talk to you about today. I will be making certain forward-looking statements, so please bear that in mind during the presentation. Glad to get that one out of the way. All right, so as we think about our pipeline strategy, and we did a recent refresh in Q4, so now we have what we describe as a de-risk pipeline of 4 clinical stage assets that either have the potential to be first in class or best in class. And the reason I say de-risked is because each of our 4 molecules are addressing a high impact, clinically and commercially validated target. Each of our molecules has shown monotherapy efficacy in the clinic, and each of those programs is addressing attractive market opportunities. And in fact, most of our molecules or programs in our pipeline are T-cell engagers, which has become an area of expertise for the company, and of course, a highly promising modality for both oncology and autoimmune diseases. And so I'll start by giving you an overview of 2 of our high priority T-cell engager programs, one in autoimmune diseases and one in oncology. And so with CLN-978, this is our CD19 by CD3 bispecific T-cell engager, which is being used to address a range of autoimmune diseases. And for this program, we have lots of data readouts this year, throughout the year. It's a highly differentiated molecule and a potential disease-modifying treatment regimen for a broad range of autoimmune diseases. We have a global development program in 3 high-value indications: RA, lupus, and Sjogren's disease. And we plan to be the first company to report company-sponsored data for a CD19 T-cell engager in autoimmune diseases. So if you follow the literature, much of the reported data has been either off-label use, compassionate use, case series, but not company-sponsored data. And I think the other thing I would add is we feel very good about the external validation now we've seen of this modality, specifically in autoimmune diseases. So for those of you who follow this space, if you looked at the data, EULAR last year, ACR this year, clearly underscoring the importance of T-cell engagers for the treatment of autoimmune diseases. And we also see persistent interest from large pharma who clearly have been very active with M&A in this space. So that's CLN-978. CLN-049 is a very unique, I would say, first-in-class opportunity to address a broad range of AML patients. In fact, in the previous talk, you heard about CD33, CD123. Here, we're addressing FLT3. And the advantage we have with this molecule is that it can address a broad range of patients with AML. We recently presented what we believe to be very compelling clinical data in patients with relapsed refractory AML, which remains an area of very high unmet need. From an external validation perspective, our data were presented as an oral presentation at this past ASH, and we've been selected for the upcoming 2026 Best of ASH meeting. So we feel very good about where we are. And towards the end of last year, we also received fast track designation from the FDA for relapsed refractory AML, and I would argue underscoring the clinical promise of CLN-049 for the treatment of AML. And the last thing I would say is we have a very robust balance sheet, and so we reported over $430 million of cash as of the end of last year, which gives us the resources to continue to advance these programs and generate multiple near-term and longer-term catalysts. So they're 2 of our high-priority T-cell engager programs, and I'm going to do a bit more of a deep dive into CLN-049, given that this is an oncology meeting. And let me say a few words about the disease area, AML. So in the US, every year, 22,000 patients are diagnosed with AML. So this represents a pretty attractive commercial opportunity. Now, patients with AML do have curative therapy if they undergo intensive chemotherapy followed by allogeneic stem cell transplant. The problem is the average age of diagnosis is almost 70 years old. So most patients are elderly and most patients are not fit enough to receive intensive therapy. So there's clearly an unmet need for a broadly applicable treatment that can address most patients. And as you can see, the survival was very bleak at 10% 5-year overall survival, particularly in relapsed refractory AML disease. Now, one of the things about AML is it's one of those areas where T-cell engagers or immunotherapy, in fact, has not yet been established. It's the last remaining area, large area in hematologic malignancies. And you can contrast that with lymphoma, also multiple myeloma, where we see immunotherapeutic approaches, including T-cell engagers. So that's one thing to point out. And we hope that CLN-049 will change this, obviously. I mentioned it is a first-in-class opportunity. So unlike the small molecule tyrosine kinase inhibitors that are already approved, they address only the mutant form of FLT3 through intracellular pathways. With CLN-049, we're actually using the target FLT3, the extracellular domain of FLT3, basically as a hook to exert the immunotherapeutic effect. So we can address both wild type and mutant FLT3. So this opens up the broad population of AML patients greater than 80% plus that present with FLT3. So that's one important aspect. Secondly, when you think about targets, we believe we have a broad therapeutic window because unlike CD33, unlike CD123, FLT3 is mainly expressed at high levels in blast cells and very low levels in normal mast cells and even hematopoietic progenitor cells. So we think we have a first-in-class opportunity, potential for high levels of efficacy for the broad all-comer patient population and a broad therapeutic window. So that's the theory. So let's go into what we actually presented at ASH in December of last year. This is the ongoing phase 1 study. And as of the August data cutoff last year, we reached the target dose of 12 micrograms per kilogram. And that's what we presented at ASH. So that was 45 patients. And with this program, we've adopted both single step-up dosing and double step-up dosing. And I'll explain why that's important. No requirement for baseline testing of FLT3 expression. So this was a broad all-comer patient population. And the way we dose in this program is using double step-up dosing as an example. First step-up dose is on day 1. Second step-up dose is on day 1, and then the target dose is given on day 8, and then weekly thereafter until disease progression or toxicity. This is the patient population that was enrolled in the study, so I'll point out two or three key aspects of these patients. So heavily pretreated group of patients, median of 2 prior therapies. We had patients on this study that had 8 prior therapies. Secondly, the majority of patients had received a hypomethylating agent in combination with venetoclax as their last prior therapy. That's a poor prognosis group of patients. 20% of patients had a prior allogeneic stem cell transplant. Over 80% of patients had one or more cytogenetic abnormalities. And in particular, I point you to 40 to 50% of these patients had the TP53 mutation, which is a really poor prognosis form of the disease. These patients, from diagnosis, survive for 6 months only. In terms of safety, we saw the typical type of adverse events you would expect in a relapsed refractory AML patient population with compromised bone marrow. The AE of interest was CRS, obviously because it's a T-cell engager. And there what we found was when we went from single step-up dosing to double step-up dosing, we were able to decrease the incidence and severity of CRS. So we mainly saw CRS grade 1 or grade 2. We saw 1 CRS grade 3 with the single step-up dose, but we didn't see that with the double step-up dose. And so a CRS rate of around 35%. By the way, this compares very favorably with the commercially approved T-cell engagers across multiple myeloma and lymphoma that we see in the market today. In terms of efficacy, this is what was really exciting for us. And so 6 micrograms per per kilogram is when we first started to see clinical activity. And so in patients that received at least 6 micrograms per kilograms, we saw a CR/CRh rate of approximately 25%. And then when we went up to the highest dose level of 12 micrograms per kilogram, we saw a CR/CRh rate in excess of 30%, which is very exciting for a relapsed refractory AML patient population. And durability obviously is important. In this swimmer's plot, you can see the 8 patients that achieved CR or CRh. 5 of these patients had a response duration greater than 4 months. And of these 5 patients, CLN-049 was able to eradicate minimal residual disease, which is really important in AML. And one of those patients went on to have a response that's still ongoing at more than 9 months. 2 of the 8 patients were able to achieve a CR and then go on to allogeneic stem cell transplant, which is very important clinically. So in terms of next steps, we're now at the 24 micrograms per kilogram dose, and we plan to provide a data update in the second half of this year. And at the same time, we're also thinking about 2 cohort expansions. So one in relapsed refractory AML, where we have the opportunity to optimize the dose per Project Optimus. So those patients won't include the TP53 patients. Then we have a separate cohort of patients with P53 disease. And this will include patients with newly diagnosed disease and relapsed refractory disease. So if you look at the NCCN guidelines for P53 AML, the recommendation is clinical trial, actually, because the prognosis is so poor. So we expect to initiate and complete those expansion cohorts by the end of this year. So clinical context is always extremely important. On this slide, you can see the drugs that have been approved in recent years in relapsed refractory AML. And I'll point out 2 or 3 key observations. The first, all of these drugs were approved with single-arm studies, mostly under accelerated approval. Each of these drugs addresses a relatively small subset of biomarker-driven patients, including, by the way, the recently approved menin inhibitors, which address 30 to 35% of patients. When you look at this data set combined overall, our belief, and it's pretty clear, that the regulatory approval benchmark is somewhere in the region of 20 to 30% CR/CRh rate with a duration of 4 to 6+ months. And so for us, the data that we've seen already, which by the way is in the middle of dose escalation, our view is we've clearly met that regulatory benchmark already with what we've seen so far with the preliminary data. And so this is why we're very excited about the opportunity for CLN-049. And now turning to the commercial opportunity, as I mentioned earlier, 22,000 patients annual incidence in the US. Unfortunately, most of these patients will go on and relapse, and that creates a relatively large relapse patient pool with an opportunity of $1 billion plus, actually. And then as we think about continued development, moving into the frontline setting will expand the use into a larger patient pool and an increased treatment duration. So that will drive the commercial value up to make it a multi-billion-dollar opportunity. The other thing I'll point out, as I mentioned in the previous slide, again, if you look at all the drugs that are approved to date, including the menin inhibitors, which address 30 to 35% of patients, we really have a very fragmented patient pool of relapsed refractory AML. With CLN-049, which is going to address a broad patient population, we have the opportunity to really disrupt and defragment the way we treat AML. So that's another reason why we're particularly excited about the opportunity for CLN-049 in this disease space. And in terms of next steps for us, it's both monotherapy and combination therapy. And so I mentioned we're going to be executing the single agent dose expansion optimization this year. That will give us the recommended phase 2 dose that we can then place into a pivotal single-arm study for accelerated approval in the future. At the same time, we're also planning a phase 1/2 combination study by the end of this year, which will then allow us to then run a phase 3 combination study, which will be confirmatory and also expand the use of CLN-049 into the frontline setting. And so the combination of the data we've seen to date, the unmet need in AML, the internal hematology expertise that we have in the company, we're excited about delivering this as a new treatment option for patients with AML. And so I'd like to sum everything up in this slide. So we have clearly seen very compelling monotherapy efficacy, both in terms of CR/CRh rate, initial durability data, as well as very promising activity in the patients with P53 disease. And as I mentioned earlier, we're still on the way up in terms of dose escalation, which makes these results truly remarkable. CLN-049 is a treatment that can address a broad all-comer patient population without the need for biomarker testing. And as I explained earlier, we believe we've already met that benchmark for the recently approved agents that I showed you, and that we have a clear development path that includes a single agent approach via accelerated approval. We have received fast track designation from the FDA, as I mentioned, combined with our deep hematology expertise, means we can rapidly develop this drug, very clear development pathway. And as I mentioned, a very commercially attractive opportunity, starting with the relapse refractory segment, north of $1 billion, expanding into frontline makes this a multi-billion-dollar opportunity. So both clinically and commercially, we're super excited about the opportunity we have with CLN-049, and we plan, rapid execution this year with data in the second half of this year for the ongoing dose escalation study, as well as identifying recommended phase 2 dose so that we can start that pivotal study in relapse refractory disease. And so with that, I'll stop. Thanks for your attention, and I'm happy to take any questions.